I am Flash Isaac, your favorite teacher from the future. You know, now I am back in the present because I heard that a lot of you are having issues with glycolysis and Krebs cycle. No, not when I am still here. You can't have issue with any topic. This is why I, the Flash, has decided to come back in time to take you this. Now, this is going to be a very, very interesting class, a very, very beneficial class, and you are going to learn a lot. Your life will never remain the same. As a matter of fact, if this is an empty promise, if you don't find this class interesting, this is my number. I will give you back your subscription. 8152. Now look at this. I actually uploaded the original video in the Flash Learners application, and you know, I was able to form the note. It took me 3, 4, 5 days to form the note, which is a very, very special and understandable note in the Flash Learners application. So I decided to share with you guys here on YouTube. And if you like this video, if you find it helpful, it simply means that you will find the over 80 biology questions on the Flash Learners application. So install the app and enjoy the notes, enjoy the videos, and a lot of other features, resources available for you to use offline. So I've spent months working on the Flash Learners application to build the best resources for every student in any level or any exams at all. The app is for you, plus 20 digital skills. So any resources you want, simply click on download for offline and you just enjoy it and watch videos. Glycolysis and Krebs cycle. Glycolysis and Krebs cycle. Don't worry, this class shall be as interesting as possible. Now, let me begin this way. Imagine you are at a food festival, a bustling food festival. Then you are given a very giant, a mighty prezzo. Prezzo is a kind of biscuit, a complex carbohydrate, just like the glucose in your body. So, if at a party you are dancing and you are given a giant biscuit, like I illustrated or like I said earlier, then imagine glycolysis. Glycolysis to be a culinary master or a chef, which comes to look at you. Wow, this is a giant prezzo. Okay, let me help you break it down into smaller form that you can enjoy. Because as big as this, you may not be able to enjoy it. Then you say, wow, thank you very much. That is glycolysis. Glycolysis is simply the process or the breaking down of glucose in your body into a small form or into a form that your body can use or that is needed by your body. And this form is referred to as pyruvate. Pyruvate. So that is glycolysis. The breaking down of the sugar in your body into the breaking down of glucose to form a pyruvate. And this happens in the cytoplasm of the cell. Cytoplasm of the cell. And glycolysis is the first recipe in the production of energy in your body. Now, there's something called energy conversion. We are told that the food in our body is stored in form of chemical energy, right? So the food is breaking down to give us energy for a day-to-day work. Now, during respiration, energy is definitely released, which we shall even look at later. We have aerobic respiration and anaerobic respiration. Aerobic respiration is when respiration takes place in the presence of oxygen. Oxygen is required for aerobic respiration. Now, for anaerobic respiration, oxygen is not required. But in any way, energy is being released. So, in glycolysis, glucose, which is a complex sugar, is broken down into smaller molecules that will be able to be used by the body, that can be absorbed by the body into what we refer to as a pyruvate. And this happens in the cytoplasm. And as a result, a little amount of energy is released. And this energy is released in form of ATP, adenyl triphosphate. You see? As simple as that. Now, from here, you already have a lot of questions being asked. What is glycolysis? Or what is the product of glycolysis? The product of glycolysis is a pyruvate. So you break down glucose into a simpler form that can be used, and this form is called pyruvate. Where does it happen? It happens in the cytoplasm. And what else? What is the aim? Why glycolysis? So break the food down to be usable by the body and for the release of energy. And how is this energy released? In the form of ATP. Now, cell in biology is the unit of life. In fact, in terms of the number of cells, an organism can either be unicellular, possessing one cell, or multicellular, having two or multiple cells. Now, cytoplasm is an organelle of the cell. We have a video where we described the cell structure and function. I'm sure you've already watched that video to be able to reach here. It's on the Flash Learners application, of course, the biology playlist. Glucose is a type of sugar or a unit of carbohydrate with the molecular formula C6H12O6. 6 carbon, 12 hydrogen, and 6 oxygen. Now you can actually draw glucose in different ways, either in straight form like this, or vertical, or in a ring form. So let's play with that, the structure of glucose. Glucose is 6 carbon. So when you draw something like this, you can say each point represents carbon. So here is a point for carbon. That's 1. Point for carbon, 2, 3, 4, 5, 6. Or you can do it in the form we are familiar with in organic chemistry, which is C, 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 and 6. So if we have this, then you have OH, you have OH, you have H, you have OH, you have H. So we are drawing the shape of, let's say, D-glucose. OH, OH, H, OH, right? And H. Then here will definitely be the opposite of what is here. If here is OH, here is H. And the tetravalency of carbon is fulfilled, is followed, is obeyed. A carbon atom should possess maximum of 4 bonds. So in the process of drawing an organic structure or representing organic structure, you must ensure that carbon does not carry more than 4 bonds. Now, I actually felt like I am in the chemistry class dealing with carbon and its compounds and organic chemistry. If you are offering biology, there is a chance that you will be offering chemistry as well. So I recommend that you take a look at my organic chemistry videos, especially on the Flash Learners application chemistry playlist. Now, 1 bond, 2 bonds, 3 bonds, and 4. Tetravalency is fulfilled. So if here is OH, here will definitely be H. If here is H, here will definitely be OH, or you say HO. They mean the same. So here will be H, here will be OH or HO. Then this carbon is also bonded to oxygen and hydrogen. Oxygen is divalent. What does that mean? Oxygen can carry a maximum of 2 bonds. So if I have oxygen here, I can either bond it to 2 hydrogen to have something like this, or double bond to carbon, because oxygen can carry maximum of 2 bonds. So you can draw this like this, or you simply say CHO on top. Now here becomes H. So let us count hydrogen 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, carbon 6, and oxygen 1, 2, 3, 4, 5, 6, complete. So this is your glucose. But for deep glucose, since we've taken here to be like this, at the bottom, you can simply compress here to be 1 carbon, 2 hydrogen, H2OH. So CH2OH. So let's put that arcanol here to give us something like this. CH2OH. Here can actually be CHO, like I said earlier. So it is easy to draw the shape of glucose. Now let's look at the ring form of glucose, because I am trying to make everything interesting for you. For the ring shape of glucose, you have something like this, like the benzene structure. Benzene structure, something like this. 1, 2, 3, 4, 5, 6. But in this case, it is not having the alternating double bond like in benzene. We are just trying to make this a ring. So how do we put 6 carbons, 12 hydrogen, and 6 oxygen in a ring? So we do something like this. That is 1, 2, has in, what is it called? Polygon. This is hexagon. Hex is 6. 6-sided polygon. Now, in this 6-sided polygon, one of the parts is definitely going to carry oxygen like this. O must be here. So with this oxygen has already achieved its divalency, in the sense that one bond comes here, another bond comes here. So every other here, they are carbon. So in standard formula, you don't put all this C here. You can just put straight line and cross it like that. Like this, like this, like this, and like this. Which is also works. But each of these corner will represent an element. In this case, carbon. So here is C, 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 and C. So let's start filling. Here, let's go here H. If you look at here as H, this is like 3 bonds. So carbon still needs 1 bond to make it tetravalent. So we bring another one here to have OH. If H is here, OH will be here. So that is cool and complete. Now, this OH is the same thing as OH, something like this. Is that taken? Yes, it is taken. Now, let's have H here. H, and here, let's have OH. Interesting. So here can have H here. Here is OH. Then here becomes OH. OH. Here becomes H. Then here we have H. Then here there is carbon here. We have 1, 2, 3, 4, 5 carbon. So another carbon will definitely be here. So which is this head part to give you CH2OH. So let's see if this actually works for us. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Hydrogen is 12. Carbon, 1 here, 2, 3, 4, 5, 6. Carbon is complete. Oxygen, 1, 2, 3, 4, 5, 6. Oxygen is complete. So ladies and gentlemen, I present to you how to draw simple form of glucose, D-glucose in this shape or in a straight chain or in your ring or round form. It has been said that glycolysis is the first stage of cellular respiration and it takes place in the cytoplasm, and it is breaking down of glucose to form pyruvate, right? Now, when you look at pyruvate, something comes to mind, pyruvic acid. Look at this. This is the formula for pyruvic acid with the carboxylic acid functional group. The IUPAC name for pyruvic acid is 2-oxopropanoic acid. 1, 2, 3, 3 carbon, prop. Oxygen is in carbon 2. So 2-oxopropanoic acid. This is pyruvic acid, and that is C3H4O3. So pyruvic acid is a product of glycolysis. And in glycolysis, of course, ATP is released, 2 ATP precisely, 4 in total, but 2 is invested, giving us a net of 2, which we shall see very, very soon. Now look at this. There is something called conjugate acid and conjugate base, right? If an acid donates a proton, it becomes a conjugate base. So look at this. If this pyruvic acid donates this, right, it becomes a pyruvate. So pyruvate can be said to be conjugate base of pyruvic acid. Pyruvate is, or pyruvic acid is the acid form of pyruvate. Pyruvate is simply the conjugate base of pyruvic acid. So once a proton is donated by pyruvic acid, we form this pyruvate, and this pyruvate is the product or is the final product of glycolysis. So what is the fate of pyruvate? What happens to pyruvate in glycolysis? Now let's look at the fate of pyruvate. In glycolysis, what happens to pyruvate depends on whether there is a presence of oxygen or there is the absence of oxygen. The presence of oxygen is simply an aerobic condition. The absence of oxygen is an anaerobic condition. So, if we have oxygen, this is what happens to the pyruvate that is formed from glycolysis. The first thing is, it enters the mitochondria of the cell. Simple question. In aerobic condition, what happens to pyruvate? It enters the mitochondria. Then secondly, what happens? It is converted to acetyl-CoA, so which leads to release of energy into the system in different of these forms. So what happens to pyruvate is determined by the condition, whether it's an aerobic condition or anaerobic condition. Now, in anaerobic condition, the pyruvate undergoes fermentation. The pyruvate undergoes fermentation, and this can happen in humans, animals, or in yeast and bacteria. So, if this pyruvate in anaerobic condition undergoes fermentation, in animals, it is converted to lactic acid. Lactic acid, which has a lot of uses. Then in yeast or in some bacteria, it is converted to ethanol and carbon 4 oxide. So in anaerobic condition, for yeast and bacteria, pyruvate is converted to ethanol and carbon 4 oxide. In animals or in humans, it is converted to lactic acid. This is what happens to pyruvate. Now, let's look at adenosine diphosphate and adenosine triphosphate. We have ADP, and we have ATP. Remember, all this grammar, everything we've been doing have to do with the release of energy. Simple. Breaking down glucose, at least to give some form of energy in form of ATP. Yes, in form of ATP. But look at the discrepancy, the disparity, the dichotomy between ADP and ATP. If the cell needs energy, one phosphate is removed from ATP into the system to give energy. Now, if the cell has enough energy, is fully charged, one phosphate group will be removed and added to ADP to form ATP. So you look at the process. If there is energy in the cell, a phosphate group is attached to ADP to form ATP. So ATP means there's energy available, fully charged like a battery. Now, but if there's energy requirement, energy is needed, the energy has to be released. So energy is released in form of ATP or from ATP, or one phosphate group is removed from ATP to the system, then you are left with ADP. So if one phosphate is removed from ATP, you form ADP. If one is removed from ATP, you form ADP. If one is added to ADP, you form ATP. So that is how it works. Now, a quick look at ADP is that it is a building block in your DNA and RNA. So if you look at your DNA, ATP is actually one of the building block of it. While ATP, on the other hand, of course, is made up of adenine, a nitrogenous base. ATP is made up of one adenine. Adenine is a nitrogenous base. And ATP is also made up of ribose. Ribose is a form of sugar. So ATP contains what we call nitrogenous base, which is adenine. Then that's why it is called adenosine triphosphate. It is made up of a sugar, which is in form of ribose, and it has 3 phosphates. If you say triphosphate, it means 3 phosphate, phosphorus, P in your elements and in your periodic table. So let us continue. We are not done. There are things you should never forget. The first is glycolysis is the first stage of cellular respiration. Cellular respiration. And it occurs in the cytoplasm of the cell. And it has to do with breaking down glucose, a 6-carbon sugar or molecule, into 2 parts or 2 molecules of pyruvate. Now, when you hear cellular respiration, the most direct word or the most direct opposite to cellular respiration is photosynthesis. Remember, when we were looking at the differences between plants and animals, which are the 2 living things, we said that plants manufacture their food via a process called photosynthesis. Meanwhile, animals cannot manufacture their food. So, looking at this, the opposite of photosynthesis will certainly be cellular respiration in animals. So, cellular respiration is the process whereby animals or things, or it is the breaking down of food molecules to bring out the energy stored in them. For every food you see, every food molecule, there is energy there which you can extract. But you cannot get that food stored in them or that nutrient stored in them until you break them down to smallest form. So that is the reason why cellular respiration is important. to break down that molecule or to break down glucose into other smaller molecules. So in cellular respiration, glucose is broken down. Meanwhile, in photosynthesis, which is opposite of cellular respiration, glucose is formed. Glucose is formed in the process. So here it breaks down C6H12O6. Here, glucose is formed C6H12O6. So this takes place in the presence of sunlight, where plants, because of the presence of the chlorophyll, make use of carbon dioxide and water to produce their own food, which is called photosynthesis. Now, this is cellular respiration is referred to as catabolic. Non-cellular respiration or photosynthesis is also referred to as anabolic. Catabolic and anabolic. Catabolic process and anabolic process. In cellular respiration, oxygen is needed and carbon dioxide is given out. That is why we say that animals, they breathe in oxygen and they breathe out carbon dioxide. In photosynthesis, carbon dioxide is needed. It's an input, it's a reactant to the process, while oxygen is an output, it is given out. This is why we say plants breathe in carbon dioxide and they breathe out oxygen. So this is actually the highlight or the comparison between cellular respiration and photosynthesis. Food is ready. We started from the bottom, now we are here. Let's look at the phases involved in glycolysis or the steps involved in glycolysis. Now, if we are looking at the steps involved in glycolysis, we'll simply say step A, B, C, D, E, F, G, H, depending on what you are looking at. However, if you are looking at the phases, we break the glycolysis process into 3 phases. We have the energy investment phase. We said that glycolysis is the process whereby glucose is broken down to give us energy, right? To form pyruvate. In as much as the process of glycolysis is to produce energy, energy will first have to be invested. Now, sometimes I wonder, why is it that after eating, I first get tired or weak before I get the energy that the food is supposed to provide to me? Is it because I need to first invest energy? Because even in business, if you need to invest money to get money, you use money to get money. You don't just get money like that. So energy has to be invested. After that, we have cleavage. It has to cleave. Then we have energy payoff, which means you've invested. It is time to reap. So the 3 phases of glycolysis are energy investment phase, cleavage phase, and energy payoff phase. So these are simply the steps, the ones under the phases. Now, the first step in glycolysis is glucose. Glucose is the subject. Glucose is the main focus, is the beginning, is the alpha of glycolysis. So in the energy investment phase, glucose is phosphorylated to glucose 6-phosphate by an enzyme called hexokinase using 1-adenosine triphosphate. 1 ATP is spent. Now, look at this. Phosphorylated means adding a phosphate group. That should be PO3 2 minus. We know phosphate group PO3 PO3 2 minus. Phosphorus. So glucose is, this phosphate group is added to glucose. That is what we call phosphorylation, right? Now, when a phosphate group is added to glucose, what the result is glucose 6-phosphate. Now, what actually happens here? The phosphate group is attached to the 6th carbon of the glucose at the top. That is just the big grammar here. We're adding phosphate group to glucose. And to do that, we need an enzyme. It does not happen on its own. It needs to be accelerated. Enzymes are organic catalysts that help to speed up the rate of reaction. If I say, John, go and get me bread, John will definitely go if he is under my authority. How about a situation where I say, John, go and get me bread, buy 2, take 1 for yourself, or if you come on time, keep the change. John would have to run. I need to keep the change. So John is motivated to do that, which at that specific time he will come back being motivated. On the normal case, he may not even come back that early. So if he doesn't come back that early, hunger may kill the person who needs the bread. So for it to occur, for that reaction to be able to achieve, we use that motivation. These are the roles of enzymes. So they just speed up the rate of reaction, or catalyst. And how do they work? I think they lower the activation energy. So if you have to go up to achieve that reaction, they just reduce that energy you need to carry out the reaction so that it will be possible. So this enzyme is called hexokinase, and 1 ATP is used. Remember, we are in energy investment phase. So we first of all invest 1 ATP to convert glucose to glucose 6-phosphate. Then after that, this glucose 6-phosphate that is produced is isomerized to fructose 6-phosphate. In chemistry or organic compound, this is what isomerism is. It's actually rearrangement. It's like a mirror, okay? That's a good example, a type of isomerism. Isomerism are organic compounds. They have the same formula, the same carbon. If there is oxygen, they have the same oxygen. If there is hydrogen, they have the same number of hydrogen. Everything this one has, the other one has. But the arrangement is different. So if this is facing here like this, and this is facing here like this, same me. Everything I have, everything he has, right? The next step is simply this glucose 6-phosphate formed from stage 1 is isomerized to fructose 6-phosphate, which means glucose and fructose are isomers, the same, just different arrangement, different orientation. In this case, we are not spending ATP. So far, we've invested 1 ATP. The third step is this fructose 6-phosphate that is formed now is also phosphorylated to fructose 1,6-biphosphate by the enzyme called phosphofructokinase using another ATP. We spent 1 here, we spent another ATP. Now look at what this big grammar is about. We have fructose 6-phosphate, a glucose with phosphate on top. Now, phosphorylated means another phosphate group is added, which means we have 2 phosphate groups. Simple. Here, we added 1 phosphate group. In step 3, we added another phosphate group. What happens here is now that the first phosphate group was added to the 6th carbon. Now, the second phosphate group is added to the first carbon. That is why we have fructose 1,6-biphosphate. So we have 2 phosphates. But to add phosphate group, nothing goes for nothing. We must invest. So we invested another ATP. So how many ATP have we invested so far? 2. This is why in glycolysis, here is the reactant, here is the product. So we know glucose so far, and we invested 2 ATP. So this is like inorganic pyruvate. That is what is here. Now, moving forward, we enter the cleavage stage. In the cleavage stage, this fructose 1,6-biphosphate is split into 2 3-carbon sugars. Look at this. This fructose 1,6-phosphate is split into 2. First part is a 3-carbon sugar. The second part is a 3-carbon sugar. Now, the first 3 carbon sugar is called dihydroxyacetone phosphate. It means 2 hydrogen acetone phosphate. Look at which is DHAP. That is what you see here. Okay, it's not here yet. DHAP. And the second 3 carbon sugar is glyceraldehyde, which is glyceraldehyde 3 phosphate. Basically, arcanal and arcanones. Arcanal and arcanones. The first part is DHAP. The second part is G3P, glyceraldehyde 3 phosphate. Here, the phosphate is in carbon 3. Here is dihydroxyacetone phosphate. So, what happens to, again, this G3P is already okay. But this DHAP is further isomerized. Like I told you, they are actually the same 3 carbon sugars, the same molecules, the same formula, different arrangement. So from this stage, they actually form, or they come in different arrangement, the same formula, but this one has to rearrange to also become G3P. In essence, we have G3P here. This DHAP also changes itself to G3P, meaning the whole essence is now, or in summary, we are now having 2G3P, 2G3P. That is glyceraldehyde 3 phosphate. So what is next? If this is being done, we are okay. We are now in the energy payoff stage. So the first stage of energy payoff is that G3P is oxidized to NADH and electron is being transferred because oxidation is transfer of release of electron. So G3P is oxidized to NADH to release electrons. Yes, let me explain it now because I may not explain it when we get there. So acetyl-CoA is short for acetyl coenzyme. It is a molecule that plays a role in metabolism. It is often referred to as hub, that is HUB, a molecule because it links various metabolic pathways. Now, how is acetyl-CoA formed? Simply by the breakdown of carbohydrates, fats, and protein, and it serves as a substrate for several important biochemical reactions. Acetyl-CoA is formed from pyruvate. Remember, pyruvate is the end product of glycolysis. So this acetyl-CoA is formed from that pyruvate and is oxidized and decarboxylated in the mitochondria. Pyruvate we get from glycolysis is converted to acetyl-CoA, and that continues in the glycolysis. Glycolysis is the second stage of cellular respiration. The first stage, no. Glycolysis is the first stage of cellular respiration. Krebs cycle is now the second stage of cellular respiration. But look at it now. This G3P is oxidized to NADH, releasing transfer of electron of NAD+. As you can see from the reactant and the product, 2NADH. Now, this G3P is phosphorylated and then dephosphorylated, generating 4-ATP. This everything that happens, this G3P is further phosphorylated and then dephosphorylated to generate 4-ATP. 4-ATP. So look at this. In energy investment stage, we invested 2-ATP. Now in the payoff stage, we now have 4-ATP, which means the net energy is 2-ATP because the one you invested, you must subtract it. So 2-ATP invested, 4-ATP formed. So minus 2-ATP, you are getting 2-ATP. So the total energy you receive in glycolysis is 2-ATP, which is one of the products, output 2-ATP. Then, what else? The final product is 2 molecules of pyruvate. The final product of glycolysis is 2 molecules of pyruvate. This is it, 2 molecules of pyruvate. And remember the difference between pyruvate and pyruvic acid. If the pyruvic acid loses one of the hydrogen ions, what we are left with is pyruvate. For most persons, what they need to know about Krebs cycle is that the pyruvate from glycolysis forms acetyl-CoA, which is used in the Krebs cycle. And what is the essence of Krebs cycle? It is actually to make energy available. And in Krebs cycle, carbon dioxide is produced as a waste, and we have high energy electron carriers being produced. Then you need to know the products of Krebs cycle. 3 NADH, which I explained, 1 ATP is produced, and 2 molecules of carbon dioxide is released as waste. Now, in glycolysis, it is the first stage of cellular respiration. Then, Krebs cycle is actually the second stage of cellular respiration. Then, in glycolysis, glucose is broken down to pyruvic acid. Then, for Krebs cycle, we hold on from there. We form carbon dioxide. It is also referred to as citric acid cycle. Then, in glycolysis, the net ATP produced is 2. 2 ATP is released in glycolysis. 2 net ATP. That is what we have, because we have 4 molecules that is produced. 2 is being consumed in the investment stage. So we are left with 2. But for Krebs cycle, only 1 ATP is produced per cycle. The cycle continues. So for every cycle, 1 ATP is produced. Then I told you that glycolysis can occur with or without oxygen, either aerobic or anaerobic. Meanwhile, for Krebs cycle, it is only aerobic. Oxygen is definitely required for Krebs cycle to take place. Then for glycolysis, no carbon dioxide is released, but it is released in Krebs cycle. Glycolysis takes place in the cytoplasm. Meanwhile, Krebs cycle takes place only in the mitochondria. Then the sequence of glycolysis can actually be linear, but Krebs cycle, as the name implies, cycle, is actually cyclic. And one of the importance of glycolysis and the Krebs cycle is that they are very important for generating energy, and they provide intermediates for the synthesis of various molecules, including amino acid, nucleotides, and lipids. Now, that being said, if you wish to proceed with the Krebs cycle, this is how it happens. We have this acetyl-CoA, which I've already explained earlier in glycolysis. It joins with oxalate to form 6-carbon citrate. Now, it releases a coenzyme A by an enzyme called citrate synthase. So an enzyme called citrate synthase helps us to join this acetyl-CoA from glycolysis with an oxalate to form a 6-carbon citrate. So in the first step, 6-carbon citrate is formed. Then second step, the 6-carbon citrate form will change into its isomer, which is isocitrate. And an enzyme that makes that possible is the aconitis. Then step 3, this isocitrate, which is the isomer, will change into a 5-carbon alpha ketoglutarate. And this step can also change NAD+ to NADH. Then the fourth step, this 5-carbon produced in step 3 will now turn into succinyl-CoA. So from 5-carbon, it becomes 4-carbon, which means 1 carbon is lost. So how is lost, thereby releasing carbon dioxide. So one of the carbon lost forms carbon dioxide, and it is converted from NADH, NAD+ to NADH. In essence, in step 4, NAD+ is converted to NADH. Same thing that can also happen in step 3. Then in step 5, this succinyl-CoA becomes succinate, and an enzyme called synthetase. Then 1 ATP is made here. So this is where the ATP in Krebs cycle is produced in step 4. Both glycolysis and Krebs cycle are actually 8 to 10 steps, depending on how you wish to break them down. Then succinate changes to fumarate, and this is aided by succinate dehydrogenase. That's an enzyme. Then FAD is turned into FADH2 during this process. And the fumarate becomes malate by adding water with the help of an enzyme called fumarase. Then this malate in the process becomes an oxoacetate, starting a new cycle. And in this stage, NAD plus is also converted to NADH. So this is actually for advanced learners. Ladies and gentlemen, we can say that with this, we come to the end of glycolysis and Krebs cycle. I do hope you found this interesting. And ladies and gentlemen, I highly recommend that you install the Flash Learners application. Visit your Google Play Store or App Store to get it, to read the glycolysis class notes. Then I have some assignments for you under glycolysis for you to answer. You'll see them in the Flash Learners application. Then you also watch the videos on the app. So there's a version of the videos of the app which is different from this version you are watching on YouTube. So in fact, this video is actually a copy of that particular video and a copy of the notes in that particular app. So we have over 80 biology videos on the Flash Learners application. So if you enjoy this, you are definitely going to enjoy all those videos in the app. So get the app, activate, and you'll be fine. So if you wish to reach out to me, why not? Or concerning the app or whatever, but it should be mainly concerning the app. So like I told you, +2 3 4 8 1 6 7 9 5 1 4 5 2. There is a customer support line, but this is my direct line for my students. Take care of yourself.